Hello, my name is Michelle Finger, and I will present the G6PD deficiency in human genetics. In this case, I selected to do a drawing and like a video of me explaining it because I thought it would be easier. The G6PD is an enzyme that has the function of producing something that is called the ribose 5-phosphate, and it generates NADPH in the hexose monophosphate pathway. So in other words, it can protect the erythrocyte from oxidant stress. The G6PD gene is located in the X chromosome, but specifically in the XQ28 location. And in other words, this is a recessive X-linked inherited disease. Therefore, females that has 2 chromosome alleles and males that have 1 will have different changes in how the disease is going to be transmitted. One thing to point is that during the embryonic development, if the female child has a response to the genomic imbalance, that will cause 1 X chromosome inactivation in most females. So most females will be heterozygous or recessive types, which in other words could be called carriers. And since males have only 1 X chromosome, they can either have a normal G6PD genotypic levels, which are unaffected, or deficient or affected males that will express the phenotype of this disease. In some cases that I have to point out, heterozygous females can carry 1 allele encoding a normal G6PD enzyme, which functions normally, or they can encode another allele with G6PD deficient function. And since there's a mechanism of random X chromosome inactivation and double X alleles in women, there is a certain buffer effect or protection in the expression of a mutant gene of G6PD in women. That is why it's very rare that women could actually express the disease and the enzyme deficiency. However, there are some rare cases in which women that have an unfavorable X inactivation can have an unaffected X chromosome is silenced in most of the cells, such as homozygous females. They can present the phenotype or symptoms and as triggering factors. That is extremely rare. I believe it's less than 1% of the female population that actually have the expression of this disease. So first of all, daughters of female carriers of the X-linked G6PD disorder have a 50% probability to be carriers themselves, and boys can have a 50% to be affected or they can present the disease. Males with an X-linked disease pass the gene to all his daughters, who will be carriers if their mother is unaffected, and males cannot pass the disease to his sons because males will always pass their Y chromosome instead of the X chromosome to the male offspring. So in this case, I did this family tree of 5 generations. I would just explain 3 cases of how the disease could be transmitted. In this case, the square is presented as a male and is not colored, which in this case, he is not presenting the G6PD deficiency. Here, there's a circle that is a female, and I added that it's a carrier, and so she is a heterozygous recessive carrier with the X-linked disease of G6PD deficiency. She will not express the symptoms of the disease, but she will be a carrier. In this case, they had 3 offsprings, 1 of them deceased, 1 of them is a female, another one is a male. So in this case, in this female, she is not a carrier, and I will explain why. I did a few Punnett squares of the parents. So in this square, this male doesn't carry anything, he doesn't have the disease, but the female has a carrier. And so here's the X chromosomes of the female. She's a carrier, so one of them will have the deficiency, or the DEF, which I showed here. And a male, he is a normal father. And so once I did the Punnett square, there is a 25% that female would not be a carrier, 25% that the female will be a carrier, and 25% that the male will express the deficiency. So I circled the offsprings that were actually conceived. And in this case, there is 25% that the male is going to be presenting the symptoms. Another one that wouldn't present them, a female. And here's another female that's not presenting it as well. So female number 2 is married with male number 1. This is not part of the family. He is an external partner, and now they got married and had kids. So this male is presenting the disease. In this case, if they have offsprings, which is a female 1 and female 3 in the 3rd generation, they will not necessarily present the disease, but female 3 will be a carrier, because when the male is presenting the disease, there is a chance that the female daughter can have carrying the recessive X-linked disease. And in this case, in brother, which he has the disease, or male number 4, he has kids with female 5, which she doesn't have the recessive, which she's not a carrier or anything. And the same thing is happening as well in the female daughter. She is a carrier too. Female 3 in this family is a carrier, and female 5 is a carrier as well. The other explanation I would do is, once they have kids with the carrier and a male, I presented a very rare case. It doesn't happen as much with male 6 and female 7. So as you can notice, female 7 is encircled and colored, and that means that she is actually presenting G6PD deficiency, which is extremely rare in females, like I mentioned. But in this case, this female has the expression of the G6PD deficiency. So in this case, this male doesn't have any deficiency at all. They had 2 male kids and 2 female kids. And I presented that the male, number 8, will have the disease, because since this female has 2 X chromosomes that are affected, there is a chance that this male can be affected as well, and all the females will be carriers. And I added that a number 9 offspring passed away, because there is actually a chance that offsprings could be affected in G6PD. For example, if an infant that has G6PD deficiency could have a symptom that is severe jaundice, which is like yellow skin, and it could be present in the first 24 hours after birth. And if it's left untreated, the infant can actually develop brain damage. So in this case, this deceased offspring had brain damage, because he is expressing the phenotype for G6PD deficiency, and also because of other complications. And so the symptoms of this disease could be triggered by external factors, such as consumption of aspirin, faba beans, medications like antimalarians, Primaquine, emotional stress, and infections such as pneumonia can trigger the following symptoms, which will be jaundice, like I mentioned earlier, that is yellow skin. There could be fatigue, rapid heart rate, paleness, hemolytic anemia, enlarged spleen, and shortness of breath. Oh, and dark colored pee. How is this G6PD deficiency diagnosed? First of all, there will be a complete medical history verification and also the history of the family G6PD. And doctors may test for lactate dehydrogenase or LDH, bilirubin levels or in a blood test, a complete blood count or CBC, serum aminotransferases, which is an exam that would verify certain enzymatic levels in the liver, reticulocyte count or levels of young blood cells produced in the bone marrow, and peripheral blood smear, which is the check-in of the size, shape, and the amount of erythrocytes that are produced normally. And this will be my whole family tree. I did a few Punnett squares that I will show in a picture in my Word document, and also all the information that I just stated with its references. Thank you very much, and have a good day.